Welcome friends, today we will be looking into other aspects of drug targeting. We have looked into the novel drug delivery systems. Today we will move a bit further in modulating and designing into looking into aspects of how we can also use the drug delivery systems for targeted delivery into the body, so that the dose as well as the side effects which arise from the drugs, that is the active pharmaceutical ingredients, can be brought down. So we will be looking into various kinds of targeted drug delivery systems in this specific lecture. So the learning objectives of this specific lecture would be looking into the targeted drug delivery systems like passive and active targeting mechanisms which help in controlled drug release and enhance therapeutic efficacy. Also the biochemical interactions which influence the drug absorption, metabolism, cellular signaling and are crucial for the targeted delivery of the drugs would be looked at. Also we will be looking into carrier mediated and receptor mediated transport systems. We will also look into active target utilizers of the moieties like the ligands and surface modifications to enhance specificity. Also passive targeting is one of the opted techniques which helps in making use of the body's natural biodistribution pattern for the distribution of drug without adding any extra ligands or any extra moieties in the drug delivery system. So the therapeutic response of a drug is largely dependent on how effectively it interacts with the cell membrane associated receptors in a concentration dependent manner. So the targeted mechanism of drug delivery is basically based on the interaction of the drug delivery system with the receptors which are expressed on the cell membrane, and the more is the closeness of the drug delivery system towards these or the more is the affinity towards these receptors, the better is the targeting and hence better is the therapeutic outcome. So in order to achieve this, it is required that the delivery of the correct amount of drug to the site of action, also the control of drug input rate must be ensured to avoid fluctuations in the plasma membrane. In the conventional drug delivery systems, drugs distribute to healthy tissues causing non-specific toxicity. This undesired biodistribution reduces the therapeutic efficacy and safety. So the major limitations of systemic drug delivery systems are the rapid clearance via metabolism or excretion, inability to penetrate the target tissue, and non-specific tissue uptake which increases the side effects. So in 1902, Paul Ehrlich introduced the revolutionary concept of the magic bullet. He hypothesized that drugs, vaccines, even DNA could be selectively delivered to the diseased tissue. So this laid the foundation for site-specific drug delivery systems. So the targeted delivery must involve the engineering of drug carriers or the drug molecule itself, so that prompt and efficient in vivo response can be obtained. So the idea here is that the drug is delivered to the targeted site so as to ensure the maximum therapeutic outcome. Now the importance of targeting is to maximize the drug concentration at the disease site, minimize systemic exposure, that is non-target tissues, so that the side effects can be reduced. Also the requirements for effective targeting are that the target site must be biochemical and etiologically distinct from the surrounding tissues. It should have some selective expression of protein markers or some receptors or some enzymatic system so that the targeting can be done. So based on that, the drug must be able to localize predominantly at this specific site. So the benefits of drug targeting are enlisted in table 1. Drug distribution is site specific, as against the conventional delivery it is non-specific. The therapeutic index is low in the conventional, however it is high in the targeted. Toxicity is minimized in the targeted delivery. Controlled release is possible. Also the efficacy is enhanced against the conventional drug delivery systems. So the targeting strategies include the passive targeting, active targeting and stimuli responsive targeting. We have looked into few of these in the previous lectures. Passive targeting is based on the leaky vasculature, tumor microenvironment and direct local application. So, this is based on the physiological mechanism of the body itself. Active targeting involves receptor targeting, antibody targeted mechanism and carbohydrate targeted delivery. In stimuli responsive, pH responsive, temperature responsive, light responsive and magnetic field or ultrasound responsive drug delivery systems are used. So, the core strategies for drug targeting are: in the passive, natural biodistribution or reticuloendothelial system uptake is utilized. This is useful for infections basically involving the macrophages or the phagocytic system of the body. In the inverse strategy, the RES invasion via carrier modification is done, so that prolonged circulation, reduced immune clearance is observed. Active targeting, which is ligand functionalized targeting for cells or organs. It is highly specific and ligand receptor interaction is dependent. So, this is what it means, that the receptors should be selectively expressed or predominantly present in the target area. Ligand mediated functionalization with biomolecules, so that specific uptake into disease specific cell populations can take place. Physical, which is triggered by external stimuli, so that controlled release and non-invasive delivery can be done. Dual, that is carrier and drug work synergistically. So, herein enhanced potency and the multi-drug resistance is reduced. Double targeting, spatial plus temporal targeting, maximized selectivity and sustained release is obtained. Combination, that is polymer protein ligand hybrid systems can also be used, and precision targeting with tailored carrier modifications is possible. So, looking at each of them one by one. In passive targeting, natural biodistribution is the underlying principle of targeting. The drug carriers follow the body's physiological pathways, accumulating in specific compartments or organs. Reticuloendothelial system targeting, the colloidal carriers are recognized and taken up by the RES, enabling drug delivery to the RES predominant tissues like the liver and spleen. Passive capture by the macrophages help deliver anti-infective agents for diseases like tuberculosis, candidiasis, leishmaniasis and brucellosis. The advantages of this system are that there is no need for ligand functionalization or active targeting mechanisms, and hence simplified formulations can be prepared. The natural accumulation occurs and enhances drug concentration in the RES associated tissues, improving therapeutic effects. However, it is limited by its low specificity, that is, the drug distribution depends on the body's natural process, leading to non-selective accumulation. Limited efficiency for non-RES sites, so passive targeting may be ineffective for tissues which lack RES system. Inverse targeting, this aims to avoid the RES uptake by the colloidal carriers. So, pre-saturation of RES is done using blank carriers like dextran sulfate, or by modification of the size, surface charge, hydrophilicity or surface rigidity. Hydrophilic carriers like poloxamine 308 and poloxamine 908 extend systemic circulation by avoiding opsonization. Active targeting, this involves modification of drug carriers using chemical, biological or physical engineering. This uses cell-specific ligands, example antibodies or peptides, to achieve the receptor-mediated endocytosis. So, in this there are three orders of targeting, that is first order, second order and third order. In the first order, that is organ level, the drug retained in the organs or tissues due to size-based accumulation, example lungs, lymphatics, peritoneal cavity. In the second order targeting, cellular based targeting is done. The drug is targeted to specific cells within an organ like Kupffer cells in liver. Third order targeting is intracellular, that is, the drug is delivered to organelles like nucleus or mitochondria via endocytosis. Gene delivery makes use of this mechanism for delivery of the drug. Ligand mediated targeting, this utilizes colloidal drug carriers like liposomes, nanoparticles that are surface functionalized with specific molecular ligands. Common ligands are like clonal antibodies, which bind selectively to overexpressed antigens on disease cells. Polypeptides or proteins, these offer high specificity for certain receptors. Oligosaccharides and aptamers, these also recognize sugar binding receptors or nucleic acid sequences. Viral fusion proteins, these mimic viral infection pathways for enhanced cell penetration. This results in high site specificity, reduced systemic side effects and improved therapeutic index. Physical targeting involves external stimuli to control the location and timing of drug release. The common triggers which are used are the temperature, that is heat sensitive liposomes release drugs in tumor regions due to local hypothermia. Light or laser, this is used to trigger photosensitive carriers or to activate photodynamic therapy agents by subjecting the area wherein the delivery has occurred to a specific wavelength of light. Ultrasound, similarly, by subjecting the area to a specific ultrasound wave. This helps in enhancing the membrane permeability or sonoporation and triggers the carrier disintegration. Similarly, magnetism guides magnetic nanoparticles to targeted tissues using external magnetic fields. These are non-invasive and controllable drug delivery mechanisms. These are real time and ensure spatial precision. They ensure reduced damage to the healthy tissues. Dual targeting, this employs drug carriers that possess both delivery and therapeutic capabilities. The carriers may modulate the biological pathways like viral replication or immune response, or they act synergistically with the encapsulated drug. These are especially useful for treating the viral diseases or multi-drug resistant infections. These help in reducing the therapeutic burden, risk of drug resistance and the need for combination therapies. Double targeting, this combines two targeting principles: spatial targeting, that is which ensures the drug accumulates at the disease site, and temporal targeting, this controls the rate and duration of drug release. This often employs zero order release systems for sustained drug levels and site activated systems for localized delivery. This ensures maximized drug efficiency at the target site and minimized off-target toxicity and optimized therapeutic index. Combination targeting, this is designed for protein and peptide drugs which are often unstable or rapidly degraded. The carriers are modified with natural polymers, example polysaccharides like chitosan or dextran, synthetic polymers which can be used for pegylation for stability, and stealth. Stealth means these help in bypassing the recognition by the opsonine proteins, that is they are not opsonized and hence they are there in the body for higher time, that is they are distributed or are in circulation for a relatively longer period of time. Vesicular or microparticulate systems like liposomes and microspheres. Homing devices are attached to the carriers which help in recognizing and binding to the specific cells or tissues. This facilitates internalization and site specific release. The benefits of combination targeting are enhanced stability and higher circulation time, precise delivery and reduced immunogenicity, and broader applications in protein and vaccine delivery. Cellular biochemistry and molecular events which follow upon the delivery of these targeted drug delivery systems are: the specific and non-specific cellular mechanisms have been explored to enhance internalization and uptake of endogenous ligands or drug carrier composites. Most delivery strategies exploit cell surface receptors or epitopes through receptor-mediated bioevents, especially receptor-mediated endocytosis. Cell surface biochemistry and molecular portals are now key modules for achieving site-specific, that is spatial, and time control, that is temporal, drug delivery. Important cell surface markers like antigen determinants and receptor subunits have been extensively studied to create biochemical and molecularly guided delivery systems. So, the molecular targets are a target in a disease cell or group of cells which are identified for the treatment requiring precision therapy. Cell surface antigens are used for monoclonal antibody production and selective targeting. Cell surface receptors are critical for recognizing, binding and internalizing macromolecular ligands. Cellular oncogenes and oncogenic viral proteins also serve as molecular targets for drug delivery. Molecular targeting enhances active drug delivery governed by the specificity and composition of the targeting agents rather than just physical characteristics of the drug and the carrier. So, cell surface receptors are also used as delivery ports. These are complex transmembrane proteins which are critical in cellular regulation, signaling, growth, secretion and migration. The receptors serve as, or they basically have dual roles, which are the transmembrane transport of ligands and cargo, also signal transduction across cellular membranes. Receptor expression levels affect the extent of carrier uptake and drug internalization. So, higher is the receptor expression, higher is the transmembrane delivery of the cargo. Receptors exhibit high ligand specificity, rigid structural binding sites and differential distribution across various cell types, making them excellent drug delivery systems. So, and also they are highly specific. So, the idea of using these receptors as delivery ports is that the identification of the right receptors, if it is done, half the job of delivering the drug to the target site is achieved basically. Now, ligands as delivery and targeting tools. The ligands function as carrier surface groups that selectively target direct drug carriers to specific target cells via receptor interaction. So, the ligands are basically the moieties which go and bind with the receptors. So, these ligands are selected based on the specific expression of receptors on the target site. So, if it's cancer, the specific receptors like growth factors or folic acid, they are selected and the specific ligands to these specific receptor sites are selected. So, the ligands can be antibodies, polypeptides, oligosaccharides, viral proteins, hormones or fusogenic peptides. Now, covalent or non-covalent binding and ligand anchoring ensures proper orientation for receptor recognition, enabling specific and efficient delivery. Now, designing carriers for cell-specific delivery depends on selectivity of ligands towards specific cellular receptors, overcoming anatomical and physiological barriers which will be there en route, and after receptor recognition, internalization and intracellular trafficking, that is movement of the complete drug delivery system into the cell, are crucial for successful delivery or genetic material delivery. So, the various cell types and their surface receptors have been mentioned in table 4. So, these are basically used as a guiding principle for selecting the kind of ligands. Now, cellular transmembrane transport refers to the mechanism by which materials are internalized, trafficked and processed inside the cell through dynamic plasma membrane activities. One of the principles which are involved is endocytosis, which allows the uptake of extracellular fluid, solutes, particulate matter and macromolecules necessary for cellular functioning and therapeutic delivery. Endocytosis is broadly categorized into two main types based on the nature of the material being internalized. First is the phagocytosis, that is cell eating, that is engulfment of large particles or pathogens. Next is spinocytosis, which is cell drinking, uptake of small solutes or fluid droplets. Phagocytosis is primarily carried out by specialized cells called as the reticuloendothelial system, such as Kupffer cells in hepatic sinusoids, tissue macrophages and circulating blood monocytes. This involves three major steps. First is the recognition, initiated through opsonization, where the surface of particles is coated with opsonins, example antibodies, complementary factors, to facilitate identification by the phagocytotic receptors. Then adhesion, firm attachment of particles to the phagocytic membrane through specific receptor ligand interaction. Then engulfment and digestion. Formation of phagosomes that fuse with lysosomes, resulting in the degradation of internalized material into basic components. Pinocytosis is a non-selective process of internalizing extracellular fluid or dissolved substances into small vesicles known as pinosomes. This occurs universally in all nucleated cells. The vesicles formed transport their contents towards lysosomal degradation pathways for digestion and recycling. So, this figure 1 basically shows a schematic representation of endocytotic pathways involved with the ligands used for drug targeting. So, the first is the phagosome, next is the pinocytosis, which are occurring in the plasma, that is cytoplasm. Then there are examples of the ligand targeted delivery of the drugs, that is receptor mediated delivery. The common lectin receptors and the corresponding ligands for targeted delivery have been mentioned in this specific table, and which most of these occur by endocytosis. Now, the receptor mediated endocytosis is a highly selective energy dependent form of endocytosis, which is crucial for the uptake of specific ligands such as nutrients, growth factors, hormones and therapeutic agents. 3 modes of cellular internalization have been recognized based on kinetic and biochemical characteristics. First is the fluid phase pinocytosis, which is non-specific uptake of solutes which are dissolved in the extracellular fluid. Next is the adsorptive receptor mediated pinocytosis. This specific uptake via receptor ligand interaction enhances solute internalization. Then there is adsorptive non-receptor mediated pinocytosis. The uptake through general binding to plasma membrane without specific receptor recognition occurs in this specific mechanism. So, the fluid phase pinocytosis is non-specific and inefficient for therapeutic delivery. However, adsorptive receptor mediated pinocytosis provides high efficiency, substrate specificity and greater intracellular concentration of the target ligand compared to the ambient levels. Free drugs usually enter cells through passive diffusion across the membranes or non-specific adsorption, whereas drug carrier complexes predominantly utilize the RME system, ensuring higher specificity and better intracellular targeting. So, the examples of receptor mediated uptake include the low density lipoprotein receptors for cholesterol transport, transferrin receptors for iron uptake, epidermal growth factor receptors for growth factor mediated signaling. Viral particles like influenza virus use sialic acid residues on the host cells, and toxins such as diphtheria toxin bind to the specific surface molecules. Then the ligand mediated transcytosis. The transcytosis is a complex process allowing vesicular transport of ligands or particulate systems across a polarized cell from one side to the another. Internalization occurs through receptor engagement, leading to vesicular encapsulation and subsequent release on the opposite side of the membrane. The features involve internal vesicle trafficking, which is aided by the transcytosis associated proteins, which facilitates drug or antigen delivery across the endothelial and epithelial barriers. So, in liver, colloidal gold particles coated with sugars like lactosylated serum albumin or mannan initially captured by macrophages and endothelial cells, but later they are redistributed to hepatocytes via the transcytosis mechanism. So, both endocytosis and transcytosis are playing a role in the transfer of these gold particles. Now, once internalized by the receptor mediated endocytosis or transcytosis, the drug carrier system undergoes intracellular trafficking, which is sorting in endosomes early or late, delivery to lysosomes for degradation or recycling back to the plasma membrane. The recognition between the ligand and receptor determines carrier fate, that is recycling, degradation or redistribution. Receptor mediated endocytosis ensures site specific drug delivery and efficient therapeutic outcomes. Endosomal trafficking. So, in acidic endosomal pH, the ligands often dissociate from the receptor Receptors recycle back to the plasma membrane, while ligands are delivered to the lysosomes for processing or degradation. So, other approaches for drug targeting are the magnetically modulated drug targeting. The drug carriers are magnetized to enable localization at the target site using an external magnetic field. So, in this case, magnetized particles are basically formulated and administered intravenously. The magnetic field is applied over the target site and particles are guided and retained in the specific area. Now, this helps in achieving up to 60% of the injected dose delivery, which is very selectively accumulated at the target site. This delays the RES clearance and allows prolonged systemic action. This is also suitable for non-RES organs like brain and muscles. Now, the advantages of and disadvantages of the magnetic targeting have been mentioned. The advantages are that it requires only 1/10 of the free drug dose for the therapeutic effect. This offers controlled release from 30 minutes to 30 hours at targeted sites. This helps in minimizing toxicity in normal cells and are adaptable to various body regions. However, this requires specialized magnets or trained professional. There is a risk of permanent deposition of magnetite in the tissues. Possible focal overdosing due to uneven field gradients can occur. It involves high cost and technical complexity, which are the disadvantages of this specific delivery. Now, the magnetically modulated microcarriers are formulated. These microcarriers are less than or equal to 4 micrometer in size, which circulate freely without embolic obstruction. These are made from ferromagnetic nanoparticles, liposomes or microspheres. These localize efficiently in microvessels and are internalized by the endothelial cells. These are used to deliver muscle relaxants like pyrocurine and dialdonium selectively. These avoid systemic effects such as respiratory repression or hypotension. The magnetic liposomes are functionalized with anti-fibronectin antibodies and magnetite particles. Their applications are tumor targeting, cellular sorting via magnetism, oral delivery to Peyer's patches for proteins and vaccines. Now, magnetically resealed erythrocytes, in this case RBCs, are loaded with both drug and magnetic particles, example aspirin, ibuprofen. These are used for thrombosis treatment via targeted delivery of antithrombotic agents, inflamed joint treatment using diclofenac loaded magnetic RBCs. The magnetic emulsions are also formulated as an oil and water emulsion containing magnetite and chemotherapeutics. The application could be via the IV route, wherein the magnetic emulsion is retained in the lungs using an electromagnet. This offers site-specific chemotherapy with high retention and minimal off-target effect. Then pH and temperature responsive dual targeting. This uses pH sensitive liposomes which are composed of the phosphatidyl ethanolamine and titrable stabilizing amphiphiles. The mechanisms involve liposomes which destabilize in the acidic environment, example endosomes. The drug is released via endosomal fusion or membrane destabilization. These are used for anti-cancer drug delivery, which are localized in cytoplasmic areas, especially in tumors. These are also very good for delivery of macromolecules like proteins, oligonucleotides and plasmids, antigen delivery and genetic material transport. The monoclonal antibody based drug delivery, antibodies or the MABs, can be conjugated to liposomes which are called as immunoliposomes, also can be attached to nanoparticles or other carriers. The target are the Fc receptors via the Fc region and surface antigens via the Fab region. Examples: the idoxouridine acyclovir is delivered to HSV infected cells. Immunoconjugates are formed by linking antibodies with cytotoxic moieties. Bispecific antibodies bind to different targets. Antibody enzyme conjugates like immunotoxins or drug conjugates. These enable tumor targeted cytotoxicity via dual targeting. Polysaccharide carriers may enhance circulation and localization. Bispecific antibodies are engineered to bind both tumor-specific receptors and immune effector molecules, example tissue factors. These are used in immunotherapy for tumor targeting and diseases with defective MHC recognition. These achieve synergistic therapeutic effects. The immunotoxins combine antibodies with toxin, replace toxin's native binding domain with the antibody-derived domains. Applications are many, like target Fc receptor expressing cells in HIV therapy. Also helps in delivery of CD4 plus T lymphocytes, suppressing infections and eliminating the infected cells. Chemical delivery systems, these are advanced prodrug strategies in which the drug is chemically modified to remain inactive until it reaches the target site, and therein it undergoes sequential enzymatic conversion into the active drug. This is designed to target enzymes and are based on enzyme substrate specificity, or design target carriers. So, carrier substrate specificity is also essential here. The CDS allows targeting of almost every organ in the body. This requires a detailed knowledge of the target site, enzyme profiling, as well as biochemistry of that specific target site. Sites which can be targeted using the chemical delivery systems are eye, brain, tumor, colon, liver, lung, skin or intestine, which helps in the targeted delivery of the specific area with minimal toxicity. Thus, to conclude, the strategic design of targeted delivery reduces systemic toxicity while optimizing drug accumulation in the disease tissues. Cellular events such as receptor binding, enzymatic activation and molecular interactions significantly influence the drug effectiveness. Transmembrane transport enhances drug penetration utilizing carrier-mediated mechanism, ensuring drug entry into specific cells, improving therapeutic efficiency for challenging diseases like cancer and infections. Balancing of passive and active targeting approaches. While passive targeting leverages natural biodistribution, active targeting refines drug localization through surface modifications and ligand based recognition. So, in a nutshell, based on the body's natural biodistribution mechanism, as well as by modifying the properties of the drug or the carrier, we can design drug delivery systems which can be delivered to the target site, thereby reducing the toxicity as well as enhancing the therapeutic efficacy by non-specific and non-target site distribution of the drugs. So, thank you very much for your patient hearing. Thank you.